today Alexis is going to tell us about some computational design of self-assembling and genetically encodable nanomachines. So yeah, thanks very much, Alexis.
Great, thank you for the introduction. Can you see my screen now?
Yes.
Okay, great. Great. Well, yeah, it's very exciting to be here. Thank you very much for the invitation. So as Anthony was saying, I'm going to talk today about a project going on in the Baker Lab, which deals with trying to design de novo proteins that can self-assemble into little like nanomachines objects. So proteins that can perform function and do some mechanical work at the nanoscale. And so to do that, we're using computational methods that were developed in the lab for a while. And we're just really starting to leverage all these methods to build more and more complex systems. And so this is one example of such systems. So very briefly, I just want to remind people what we mean by computational design. So computers allow us to find a correspondence between sequence of proteins and their energy, right? And so as much as you can fold sequences into and try to find the shape that these sequences encode, we can also specify a shape of the protein and then find the lowest energy sequence that would allow this protein to exist. And so that allows us to build a lot of de novo proteins, fully synthetic proteins that don't exist in nature. And so we're really always expanding the methods we have, and now we're really starting to be able to design increasingly complex systems. And so what I wanted to do during the lab is really trying to see if we could start designing machines. And so what I mean by the machine is just a device that can change the direction and magnitude of a force. And so if you look either at what humans have made, so starting from simple machines that were designed like a long time ago, we can assemble a collection of these simple machines and build even more complex machines like cars or trains or things like that. Nature kind of come up with the same concept where like there's like little modules that have evolved to be extremely efficient at processing energy or information, and then you see this evolution of this incredible collection of machines that allow life to perform of the function of the cell. And so what's starting to appear clear is that there's an interface between the nanoscale biology and the macroscale human engineering of machines, where we can start playing around with concepts, deciding to build biological machines that, because of their substrate, they can be genetically encodable, and so that, in theory, allows us to explore really interesting biotechnological applications. And so we're starting really simple here, just trying to build very simple mechanical machines made of proteins. But the idea is that once we have a collection of simple machines, we can put them together into a more complex machine. And so what's also important to keep in mind is that macroscopic scales are very different from nanoscale, right? Because at nanoscale, you have this Brownian motion-dominated environment that's extremely viscous. And so the concepts to design, the principles to design machines on nanoscales are quite different. Okay, so here's the big picture of the project. So what I really want to achieve is try to design a simple proof of concept machine that defined as a rotor. So you have an axle and a ring that comes around it. And so this two-component system can then have a rotational degree of freedom. And so you can have rotation of one component relative to the other. And so to build this kind of system here, that's made of an axle and a ring around it, well, we have to start by designing the two components separately. So we come up with computational methods to design axles and rings, and then we design the interface between these two proteins so that we can self-assemble in solution into these systems. And so because of the symmetry of such homoligomers, then you have this kind of ratcheting mechanism in which the system can populate different states that are defined by the symmetry of the two components. So once we have this ratcheting system, then the idea is to design the interface by installing a catalytic activity so that we can bind a small molecule interface and also catalyze it so as to release some energy at the interface. And so that in theory will allow what we call the brine ratchet, which this energy from the binding and catalysis biases the thermal fluctuation and therefore will actually produce work in nanoscale. So I'm going to talk first about the computational methods we use to design the machine parts, and then I will take you through the assembly process and the actual machine characterization. So we are able to design, using computational methods, we're able to design all of these machine parts. So we first started by focusing on these axial structures, which are high aspect ratio structures. And so we wanted to first build these and then build the rings that will go around them. So I really tried to design a very large library of parts. And the idea is that the more parts we have, the more machines we can build, and the more hypotheses we can generate and test. So as you can see here, I'm showing like some proteins with their negative stain electron microscopy density around it. So we use electron microscopy to validate these building blocks after we design them. So I generated a lot of different symmetric building blocks, again, to increase the diversity of the library. So here I'm showing you a few of them. So here you have dihedral D2 symmetry, D3 symmetry, D4 symmetry, and even bigger symmetrical oligomers like a D8 symmetrical oligomer here. So these axles belongs to two kinds of symmetry, cyclic and dihedral. And then we build some rings that could go around these axles, and mostly they're cyclic symmetries. And so we made a bunch again like of different parts that are C3 symmetric to C24 symmetric, and again we try to diversify the library and cut these parts into multiple monomers and increase the diversity by fusing them systematically to other proteins. Okay, so this is just like examples of a few cryo-EM structures we got for such designs. So here you have a D8 axle, as you can tell here. So it's a protein that's made of 16 monomers that come together into that like a D8 symmetry. Here you have a D3 axle, and here are two rings, one C3 ring here and one C4 with internal C24 symmetry. And so as you can tell, the design that comes out of the computer is actually really close to what we find in a tube, right? Okay, so then I designed all of this big library of machine parts made of ring and axles. And so the idea was then, once we validated them, to design interface between them so that they can come together in solution and assemble. So we systematically take axle and ring. And so what we do is that we try to thread the ring around the axle computationally, and then we sample the rotational and translational degrees of freedom. And then we design the interface between the two so as to like minimize the energy of binding. Okay, and so what we end up with is like a collection of these routers in the computer, and then I went ahead and like synthesize them. And so here I'm going to show you a few of these complexes. So here's an example of a D8 axle in gray with two rings that are C4 that bind around it. So here you have the model in the computer, and here this is a cryo-EM structure of this structure. And so you can see that we're very happy with that because obviously the structure really matches very well the model. And so interestingly, we can compute here. You can see like this is an energy map. So if you rotate the rotor around the axle, you get Rosetta can compute an energy that tells you about how likely the structure is going to be. So what you can see here is that the energy landscape is very rugged, which means that the rotor is going able to populate all of these very low energy minima and jump around in these lower energy states. Here are other examples of such rotor complex. So here we have a C3 axle with a C3 ring with a cryo-EM again here. This is a D3 axle with a C3 ring around it. Again, some very nice cryo-EM. And here you have a D2 axle with a C5 ring. And so interestingly, we found that for some of these systems, the cryo-EM could show us some structures, but we are having a very hard time trying to refine the data because the parts are moving a lot, and so we get this kind of like blurry pictures of like the whole complex. And interestingly, there's a correspondence between what we see in cryo-EM and the actual energy that Rosetta is able to compute. And very briefly, if we look at this design here, so that's an example of such a behavior. So Rosetta can really capture the energy of the system, so we can actually compute the landscape of what the system is going to populate. And so we found that looking at experimental cryo-EM data, we're able to find these lowest energy states that Rosetta can compute. So for example, here, this C8 symmetric minimas along the rotational axis, they look like this, and that's actually what we can see in the microscope. But then you have these other minimas that are much less energetic, but they can happen and can also resolve them using the microscope, but then much more rare. So we're very interested because it seems like we could really starting to have a picture of like what Rosetta can see, predict, and what we can actually get in the tube. So the next step was really to try to look at single molecules in solution and see whether or not they can actually rotate. And so the experiments that we came up with to do that is to attach the rotor complex in a microfluidic chip and then use a microscope, a fluorescence microscope, to track the displacement of the nanoparticle attached to the rotor. And so this experiment can allow us to monitor in real time with sub-nanometric accuracy the displacement of the nanoparticle attached to the rotor. And so that's the kind of plot that we get from this experiment. So here you have distance and in nanometers and distance in nanometer. And so this heat map is the center of the bead recorded over 20 seconds. And so we obtained this kind of donut shape that we're very happy with, that show us that the nanoparticle is able to diffuse on the surface, but only in a donut shape, which means that it follows the constraints imposed by the axle of the rotor. Okay, so then we went ahead and measured a few of these rotor de novo design protein rotors. So this one is a C3 axle with a C3 ring, and here I'm showing you the states that nanoparticle can populate, and you can see that interestingly it's like C3 symmetric, which means that it seems like the bead can populate C3 symmetric wells, which actually corresponds to the symmetry of the design. We can also see that if we look at the population of such states, it follows this, again, it's like C3 symmetric shape that actually corresponds to what Rosetta can compute again. So we're very happy with that. So this is still preliminary. We're collecting a lot of data, but it seems to show us that we can design such structures to rotate in solution according to design. So here are other few examples of different symmetries. And again, you can find this like rotary behavior. And it seems like the energetics of such rotation also behaves accordingly to the computed energy. Okay, so I just showed you pretty much that we could design a structure. They can self-assemble, they can rotate. And now, so they can rotate in a free brane diffusion way. So there's no input of energy. There's no work being done. It's just really just a simple machine. But the next step of the project is to actually input biochemical energy into the system so you can bias rotation and then generate work. And so that's what I'm going to talk about here. So the idea again is to install a catalytic activity at the interface between the two components so that you can bind and cleave a small molecule and release energy. And so very briefly, the theory behind how these kind of biological motors work is what we call the Brownian ratchet mechanisms. And that's how natural motors work. So very briefly, what happens is that if you manage to have a system that can ratchet between different states, then you can bias the population of these states and the direction in which it will ratchet by binding a small molecule and releasing energy. And so that's what I'm depicting here. So if you have an asymmetric potential, so that is the rotation along the rotational axis, if you look at the energy, you need an asymmetric potential like this, and then every time you bind a small molecule and go down, you increase the probability of the whole system aligning into one direction relative to the other. And so that's the mechanism by which you can generate work. So we went ahead and used these principles to try to design rotary motors. So what I did is that I took these designs I made that I knew rotated, and then I designed I installed de novo enzyme at the interface. And so the molecule that we targeted as a fuel, right? So molecule that can bind, cleave, and release energy. We use this methyl group that we know how to design computationally catalytic groups that we can install in backbones. So this reaction is energetic enough that it's just slightly above the thermal threshold. So in theory, it should be enough to power rotation. So again, the reason why we chose this fuel is because we now have some experience in designing such de novo enzymes for this kind of reaction. And so we went ahead, made the proteins, and then added the substrate, and then measured the product generation. And we could see that for some of these designs, we could actually get some activity, which was really promising. And interestingly, we can also use modification of such substrates where instead of this like alkyl group, you actually have a diketone. And that's very useful because this diketone actually reacts covalently with the protein and generates an adduct that then we can measure. And so I'm going to show you why this is also very interesting, because by covalently reacting this product on the protein, then you can modify the energetics of the interface and actually lock in place the protein, the rotation of the protein by modifying the interface. Okay, so this is very preliminary experiments that we're doing, but they're very promising, so I'm going to show them to you now. But we have to do a lot more work on this. So we went ahead and really like took these de novo design rotary motors and added fuel or inhibitor to them and measured their rotational properties. So what we do here is that we put this protein on the surface, right? And then we measure like we track the nanoparticle attached to it. So then we add some fuel, then we measure the rotation, and then we add some small molecule inhibitor and measure rotation. And here I'm showing you what happens. So this is one molecule, and this is the number of revolution for 10 minutes. And so this is just Trees buffer. So you can see that the molecule can go in one direction, the other direction, one direction, the other direction. And so when you sum the whole number of revolution, you actually are at 0, right? And then you add some fuel. And what we saw is that it seemed like preferentially, we slightly biased the probability of rudder going one direction relative to the other. And so you end up with this like delta of revolution, which is around 300 in 10 minutes. And then when you add this suicide inhibitor that covalently reacts with the active site, then what we saw is that we could lock in place the rudder. I'm not going to go into too much detail, but we could track the molecule, and it seems like it's allowed to move by a discrete step, which is actually consistent with like this like C3 kind of behavior. And then again, like Rosetta can capture the energetics and seems to be aligned with the experimental data we have. Okay, so that's what I wanted to show you, but very briefly, so we're thinking about other kinds of designs and other machines we can make. And the first reflection I had, especially during COVID, since I had a lot of time to think about these things, is that the thing I designed is really just a very simple scheme, and like there's like 3 binding sites that are symmetric for the fuel, and that's really definitely not the most efficient way to do it. If you look at nature, for example, if you look at ATP, you have these really complex coupling and conformational changes between inside the fold between the whole like active sites. And if you also look at what man has been able to engineer, you can see that these engine designs are usually very complex and also have some very complex coupling between parts. And so that's something so during COVID, I had the chance to design a few new prototypes, and so that's one of them where I have some very preliminary data. But the idea here is to reinstall a quasi-symmetric fold inside. So the idea is that you have 2 components that are never exactly satisfied, and so when you have this binding site, you create this kind of constraint that allows only binding site to be open, and therefore the idea is that it might increase the efficiency of such models. Something I've also been working on is trying to install light activable behavior, where instead of like installing a catalytic activity where you bind a fuel and cleave it, we can also bind a small molecule that can be activated by light. So by which you change the formation of the molecule by shining light on it. And so that also should allow to power a rotary engine. And so we have primary data on that. It seems like we're able to bind the small molecule. And then last, I just wanted to say that we're thinking about slowly starting to think about building compound machines. So in the same way we do for macro scale complex machine, we can start like putting pieces together of the simple machines we built to try to make more complex things. And so these are such examples. So here we're just coupling two axles to have two movable parts in the same systems. And we can also think about building gears. So very simple protein systems that are safely coupled. All right, so I hope I convinced you there's still a lot to do at the nanoscale, and especially with proteins. And on this, I would like to thank all my collaborators, and especially David Baker in the Baker Lab, my mentor, which always has great ideas and helped me a lot with all these projects. A lot of people in the Baker Lab, my collaborators in the Coleman Lab, especially Jesse Hansen, we did a lot of cryo-EM. Zev Bryant and Kevin in the Bryant Lab at Stanford, we did single molecule experiments. And Phil Bradley at the Fred Hutch for really cool designs. Thank you very much. And if you have any questions, I'm happy to answer them.
Okay, thanks Alexis for a fantastic talk. Anya, I'll hand over to you for questions.
Perfect. Thank you so much. This was amazing. We have a question from Roberto Chica, and now I have a right to unmute Roberto. So maybe you can ask the question yourself. So Roberto, would you like to unmute yourself and ask questions, Alexis? Roberto, we cannot really hear you. So anyways, I'm going to read out the question. Beautiful work, Alexis. Does the number of revolutions in 10 minutes in the presence of fuel match the K-cut of the enzymatic reaction?
Oh, I'm sorry. I think I could not hear you very well.
I'm sorry. Beautiful work. Does the number of revolutions in 10 minutes in the presence of fuel match the K-cut of enzymatic reaction? Yeah.
So again, sorry. So these are preliminary data. We're still working on that. We're not sure exactly. So it's very low efficiency, right? Obviously, and we don't know yet exactly how to make sense of this data. So I'm not sure I can comment anything useful on that. So yeah, and so it's quite hard because the setup of these experiments is such that we, first of all, like the way it's all set up, so like the protein is isolated on the surface, and then you have a giant bead on it, so that also like puts a lot of stress on the system. So it might be really hard to have a good correspondence between the biochemical data and the rotation. But we'll get there, and we'll have more data soon. But I'm sorry, I cannot comment very specifically about that.
Thank you. Another question from Zach Wu. Really exciting work. Can you talk a little bit more about the design constraints for the enzymatic driving force? For example, how was it picked?
Mm-hmm. So the reason why I picked this reaction is, first of all, it's like the energy release is slightly above the thermal threshold, so that was the first reason why. But the second is that it's one catalytic activity reaction for which we have a lot of experience computationally to install in de novo scaffolds, and so that's why I really focused on this one. We could use a lot